Hello everyone, we're here today to talk about transgenic organisms. I'm going to go through what these are, how they can be used, and a few examples of technologies on how to make transgenic organisms. Basically, these are organisms that have had their genetics changed in some way, and so they are bioengineered genetically. The first example that we're going to talk about is transformation, which is a way of adding genes to bacterial genomes. Then I'm going to talk about supercorn, which is a GMO, a genetically modified organism, and one of our crops, and we can make it so that corn, etc., grows with different properties, like, let's say, pesticides. The third thing I'm going to talk about is knockout mice. Knockout mice are a huge tool in cancer research and other biological fields in which we have mice that are missing certain genes. I'll finish it up with a hypothetical look at a permanently spicy tuna. Transformation of bacteria is relatively simple. Bacterial DNA is held in what's called a plasmid. It's a circular piece of DNA. So unlike humans, where our DNA is wound into chromosomes, it is just one circle of double-stranded DNA. That allows us, and it's pretty small, they're usually around 5,000 to 6,000 base pairs, and so it allows us to use that plasmid in order to generate new DNA. And so we have this plasmid, we can edit the plasmid, we can synthesize a full plasmid, and then we can insert that into the bacteria. Bacteria will uptake the DNA, and then they will express the new trait or traits from the new genes that we put into the plasmid. So plasmids are these circular chromosomes. And basically, each plasmid has a certain number of genes on it. And we can engineer plasmids. This is a really common one called PBR322. It's about 4,000 base pairs, like I said. And it contains two real important genes, one for ampicillin resistance and one for tetracycline resistance. And so we can make bacteria that are resistant to tetracycline or ampicillin using this plasmid. A bacteria that has uptook this plasmid will be resistant to both of those antibiotics. This ORI is the origin of replication. This is important. It's where kind of our RNA polymerase is going to bind and start replication. And so in order for this gene and this gene to be turned into mRNA, as we learned about in the central dogma, it has to start somewhere, and it's going to go in this direction. Another awesome tool that we have is something called a restriction enzyme. A restriction enzyme, or a restriction endonuclease, is an enzyme that attaches to DNA at a certain double-stranded sequence. And so we can see here we have GAATTC and its complementary strand on the other side. EcoR1, one of the most common restriction enzymes, recognizes this sequence, and it will cut the covalent bond between the G and the A here and here, and it will break the hydrogen bonding intermolecular forces between the two strands here. And so what we'll be left with is a plasmid that's sliced open. We can use this to our advantage. So if we were going to use EcoR1 to cut this up and NDEI, then we could cut this plasmid in half. And so what we'd end up with is this piece that codes for tetracycline resistance and this piece that codes for ampicillin resistance with the origin of replication. And so we get those two, we cut here and at the bottom, and we get these two halves. And so we've cut it into two 2,000 base pair rings. We have this side with ampicillin and the origin replication, and we have this side. This side isn't super useful because it does not have the origin of replication. And so we're just going to use this side. And what we can do is we can then attach genes that we are interested in studying. And so we can attach a DNA sequence that is a promoter region. We just talked about gene regulation. And so a promoter region would be where something would bind that would activate this gene. And so we put in a promoter region, which allows, and then a gene for a certain protein, and this would allow us to transcribe and then translate these genes into proteins. And the bacteria would be ampicillin resistant. And so we would be able to tell that our plasmid was inserted into the bacteria because they would grow in the presence of ampicillin, along with expressing those genes that we wanted. A really good example of this is the pGlow experiment. This is a lab that we would usually do in BioA, but we have done away with this year because of timing and the online setting. And so this is the plasmid that we would usually transform into our bacteria. And so you can see that this plasmid has a few different genes. It has beta-lactamase, which is the ampicillin resistance gene. It has the GFP protein. GFP is actually a protein from jellyfish. What is it doing in a bacteria? It is going to make them glow. And so it's a little fun application where we can see the proteins glowing at the end. Then we have this gene here, which is our promoter region. And ARA-C stands for arabinose. And so what this means is that when there is arabinose present, then we will transcribe the GFP gene. That's important for a couple reasons, which I'll get to in my next slide. First, I want to talk about how the transformation takes place. And so we're engineering this plasmid, and we want the plasmid to go into our bacteria. How we do this is we basically kind of brutalize these cells. We poke holes in them, make them what we call competent. And competent E. coli cells or bacterial cells are ones that just have holes in them and so are going to take in this DNA. And so they'll take in the DNA, they'll take in the plasmid, and that plasmid will be integrated into the DNA, or it will exist on its own. Bacteria can have multiple plasmids within them. Another way that bacteria transfer genes, and something that's helpful for us, is that this bacteria can form a link with another bacteria and pass an entire plasmid along. And so once we have transformed into one bacteria, it will spread pretty quickly by this route. So if we go back to that pGlow experiment, this is why the arabinose promoter is really important. If we grow cells that did not get the pGlow gene here on LB, which is where bacteria grow, they will grow. Right? We haven't done anything to these bacteria to make it so they won't grow under normal circumstances. So yes, we get bacteria growth in this. If we take bacteria that has not gotten our plasmid and we put it on a plate with ampicillin, then we will be able to tell that it doesn't have our plasmid because it will not grow. It will not have that gene for resistance to ampicillin. So no, we will not get any bacterial growth here. If we take bacteria that have been transformed with the pGlow plasmid and we plate it in normal Loria broth, which is what bacteria like to grow on, and ampicillin, then we should get growth in the presence of ampicillin because they should have that ampicillin resistance gene, that beta-lactamase. And so yes, we should see growth. But they won't glow. And they won't glow because there's no arabinose. In this fourth trial, we have cells transformed with pGlow that are in LB, there's ampicillin, and there's arabinose in the gel. And so here we should get growth and it should glow. And so this is a fun visualization that we normally do, but it's only one application of transformation. There are a world of possibilities out there. My senior year in high school, I was in a biochemistry research class, and we were introduced to the idea of transgenic bacteria by creating bacteria that metabolize a certain alcohol and ended up smelling like bananas. It was wild. Some might say that it was bananas. But we were basically growing bacteria that when we fed it isopropyl alcohol, it smelled like bananas. And if you've ever worked in a lab with bacteria, that is a very good thing. This has also been used for maybe more useful things, more important things, like creating insulin for people with type 1 diabetes or type 2 diabetes. And so Novolog and many other companies that produce insulin use bacteria to kind of harvest the protein. And so they'll insert the insulin gene into a plasmid. They'll grow up the bacteria, they will express that gene and produce the protein, and then you can kind of grab the protein from the bacteria and harvest it and get it ready to put into humans. You can also use it for some fun applications. So in that class, my senior year in high school, my group was designing bacteria that could sense the presence of mercury in water. And so the promoter region was like the arabinose in the piglo experiment. It was activated by mercury. And so when this bacteria came into contact with mercury, it would glow. And so we used the same thing as the piglo, but it was activated by mercury, and so it would be really good for sensing mercury in different water populations where we might want to go fishing, etc. So that is transformation. Up next, we have GMOs, and I'm going to talk about one specifically, which is supercorn that is pesticidal. And so basically what happens is we use transformation. We grow up bacteria that have the plasmid that we're interested in, we transform it, and we kind of create a lot of copies of the desired segment of DNA using the bacteria to do that. We then put the bacteria in the plant cells, and they will receive the DNA from that bacteria. And so when those plant cells that we're growing in vitro, so in a petri dish, when those grow up, they will produce seeds that have inherited that trait. When we plant those seeds, we will get corn that actually produces its own pesticides. And so the seeds are planted, the crops have inherited the desired genes, and when they grow up, they will be pesticidal all on their own. We do this with a lot of different crops. Some examples are corn, soybeans, cotton, canola, squash, sugar beets, grapefruit. But we use this in a lot of agriculture because it makes growing and producing food for a large amount of people really easy. GMO has kind of a bad rap, but really it is just genetically modified. It is not grown in any bad way. It cannot harm humans. It just expresses different traits. The last thing I want to talk about is the third thing I want to talk about is knockout mice. And so knockout mice are mice that have had genes removed. And so basically what we can do is we can take mice, mouse embryonic stem cells. So these cells can become anything in the mouse's body. And we take these cells and we mess with their DNA. And then we insert it into a blastocyst, which is the precursor to an embryo in a mouse. And we will get a mouse that expresses some of these things. For example, before my senior year in high school, I worked at Dana-Farber Cancer Institute, and we worked with P53 mice, which are mice that have a tumor suppressor gene removed. We basically replaced that gene with an inactive version. And so the mouse gets tumors all the time, which is really helpful when you're studying cancer. The last thing is this hypothetical spicy tuna. So we have chilies, and we have isolated the protein that makes them spicy. And so what if we could take this protein and the genetic sequence that codes for this protein and make tuna express it in their muscles, in the meat that we like to eat? Kind of the result of this would be permanently spicy tuna. And that would be amazing because it would make some great sushi. And so we can take this gene from chili peppers, put it into tuna, and get spicy tuna. Because why not? And so we've gone through all of these things. And now what we want to do is it's your turn. We're going to do a project in which you're going to design a transgenic organism. In homework 5.7 for tonight, you'll fill out your project proposal and create something that helps people in some way. Have fun with this. Enjoy.